Hello, everybody. I'm Steve Becker,
Dennis Warnicki here, Tanio Biologicals.
here to have some fun and talk about, kind of what we do, and what we do is microbes.
Yeah,
And microbes, as we always say,
are good.
microbes good.
Microbes good.
So the title today, Understanding and Utilizing PGPR for Enhanced Soil and Crop Performance, because that's what we really need to focus on is how do we maximize our plant's potential? How do we maximize what we can get out of the plant, what we can get out of the soil, what we can get out of our yield, and how do we make that a nutritious product, a nutritious material for the consumer?
And as we say, a picture is worth 1,000 words.
At least
And yeah, and maybe more.
2001.
And we love to see pictures like this. We talk about this all the time, and we talk about the roster roots and everything that's going on and how we love to see that. So we're going to break down today what all is going on basically in this soil profile, well beyond, we so often talk about nutrition. We're going to talk well beyond what's going on here based off of nutrition, based off of plant expression, genetic expression, all those things, carbon,
Yep.
all of those things is what we really want to talk about.
Exudates, humification, all of that. And that's where we're going. The topics today, we're going to talk a little bit about carbon, soil, humification. Biology is the foundation. I have emphasized is, because we're going to talk about that and how critical these systems truly are and how interconnected they really are. Program, not product, which is what we always talk about. Unlocking potential and what those functions of the PGPR, plant growth money rhizobacteria, are. Nitrogen.
We're going to beat it up again.
Yeah, no,
we always do.
We got to pick on nitrogen, but I mean, for good reason.
Yeah.
For very good reason.
And we're going to talk about some exciting stuff when it comes to nitrogen.
We are, because there are benefits, there are things that we see going on, and there are changes and shifts in a lot of the psychology and a lot of how people are approaching some of this, which I think is a good thing.
Yes. And how we think about it.
Yeah.
Because really everything in this universe is connected to everything else. You cannot isolate one mineral, soil, microbe, plant, animal, or human component and treat that component as the others don't exist. Once you gain an understanding of the interconnected or interrelated character of all things, and what I want to really mention here, we sometimes, when I'm speaking with a grower, it's like, well, I need a microbial product that I can apply to help my crop. Well, if we apply a microbial product, but we don't think of the nutritional aspect, that foliar nutrition, that soil nutrition, or we apply a microbial inoculum and we don't think about the human component, tilling, herbicides, fungicides, insecticides, soil sterilants, all of these things that are interconnected when it comes to agriculture, because they're all part of the agriculture process. We talk all the time. Potatoes, how do you get them out of the ground without disturbing the peanuts? I was thinking about developing a potato and peanut tree.
Ooh, that'd be. Can you also grow your candy corn on
it? Candy corn, I've already got that going. But it's that missing piece, that puzzle piece that we're talking about. What is that missing piece that gets us through the biology?
Well, and thinking about it like a puzzle, when in a reductionist view, when we're trying to isolate any one of these things and focus on it, we're missing the big picture. So we have to, like Dennis was just saying, look at how all these things are interconnected to create that overall picture. And when we do that, we start to see this sort of connection, these sort of cycles. And that's what all of this is about, cycles.
Well, and biology is what we call the foundation of nutrient availability. All of these things that we see, they talk about the 18 essential nutrients. It basically in this picture, all of these things are needed by the microbes. They're needed in the soil. They're needed by the plant. They're needed for growth. They're needed for reproduction. They're needed for enzyme production, photosynthesis, nitrogen conversion, protection, secondary metabolites. All of these things are needed in the plant, and these same things are needed for the human body.
Crazy.
It's crazy how these things are connected. And so often we separate these things when we talk about it. A lot of times we talk about human health, and we don't start at the foundation, which is the soil and the microbes and how that all functions within that environment. And, a lot of this is not even listed here when we start to talk about the things that maybe aren't essential, but we talk about nickel, we talk about cobalt, we talk about some of these things that
iodine's not on there,
that aren't even listed here. These are all part of plant nutrition, human nutrition, and microbial availability and microbial nutrition.
All part of the puzzle, if you will.
Exactly. But how does nature do this, Steve?
So when we're looking at it, nature starts off earth, rocks, volcanoes, all of this is made up of everything. When we look at what rocks are, that's what we start off with, our parent material, our bare rocks. The first organisms that are going to start breaking that down, they're bacteria. They're called chemolithotrophs. They produce enzymes and acids to break little tiny microscopic bits off that they can then consume the individual minerals out of. So that's where the mineralization, that's where minerals initially start their journey into life. So we have these bacteria that are starting to break it down, making a little bit of nutrition available, which then allows lichens. And then after the lichens, we start to see small annual plants. And the lichens, we start to then move into grasses, perennials. We start to see increasing complexity. And that's what we really need to focus on, is how can we maximize the diversity, the biodiversity, and the density of different organisms that are associated with these systems. And this is called primary succession. And this is the idea of increasing our complexity until we have an apical or an apex or a climax community, which is typically a forest. And forest is full of all sorts of different organisms. That we have them breaking down material, we have them going through humification, we have them digesting them down. So there's a lot going on there, and that's if we allow nature to do its natural action. And this, we're talking about hundreds to thousands of years. The downside of that is anything we can build, we can also degrade and destroy. And if we're utilizing the improper practices, we start to see this go in the reverse, where instead of being able to grow these more complex communities, these grasses, perennials, we start getting back into these smaller annuals. And then we start talking about the word that we're not supposed to use anymore, weeds. Weeds are just plants that are doing a job that we don't want them to be doing, but they're doing it on purpose because they're trying to increase the complexity of the system. They're trying to increase the nutrient availability. They're doing a job for us.
Well, and so often in agriculture, in primary succession, we are moving backwards
Yeah.
in a lot of scenarios.
Yeah.
That's just part of agriculture. And a lot of times we talk about weeds based off of accumulating certain nutrition, but they're also there to recruit certain microbial communities.
Absolutely.
Based to create those enzymes to make that nutrition available that is locked up in our soil environment. So all of this primary succession is, how do we utilize that? What things can we do in agriculture to speed up that process? And sometimes, I guess, if we look at that, we look at, how does nature do it? Biologically.
Yeah.
How can we do it? We just speed it up biologically. And we have to think about this. The biology is just not in the soil. It's just not in that rhizosphere, that rhizoplane. We have them surrounding the root, the flower, the fruits, the stems, the leaves within the plant tissue, surrounding the seed, inside that seed, on the outside of that seed. All of these are part of that biological process, which is agriculture.
A complete system, and that's what we have to look at. And I like this sort of picture because we're looking at this idea of excuse me, plants and humans having a very similar concept. We have a host genome, our genes associated with us. But then this idea of the holobiome, the whole organism, the hologenome, and that's including all the organisms that are associated with it. And we see so many different functions going on. We have massive amounts of communication going on inside of our body, but also around the plant.
Well, and if we think about, we go back to that picture where we talk about the soil and the microbes and the plant and the human, and we look at that based off of the digestive system. Plants are not great at extracting nutrition from the soil. They need that microbial community. The human body is the exactly same way. We need that microbiome in our digestive system, basically for nutrient availability, our immune system, all of these things that function. Really, the similarity is actually very exciting.
Well, and I mean, part of that is because all of this is life, and life requires pretty much the same basic recipe. And the goal of life is to spread itself. Bacteria want to make more bacteria. Fungi want to make more fungi. So they're going to do everything they can to create an environment that is conducive to that. So if a bacteria that is growing off of the exudates of a plant can increase the exudates from that plant, guess what it can do? It can grow more. And that's its overall goal. So we start to look at these basic equations of what is life? What are the requirements of life?
Well, when we start talking, Bruce had said this a long time ago, with the exception of light, The soil can supply all of these things to
Yeah.
the plant. The heat, the air, the nutrients, the water, all of these things they can supply to the plant through the soil
Hmm.
because they're everything that that microbial community within that soil environment needs to exist. So when we start to look at this, so often we go to the law of minimum and we say nutrition is our greatest limiting factor. Even if we have all of our nutrition, but we have soil compaction and we're
Hmm.
lacking microbial digestion, we have saturated soils, we're lacking water holding capability. All of these things are, as Bruce says, interconnected.
Yeah.
And we cannot have optimum growth and optimum yield without these other things. So we
It's
have
always a
to pay
balance.
attention to both. It's a balance. And it's amazing how these two basically support each other. And so often when we look at that, it's actually really interesting because we think of who are we feeding within that soil environment. And if we think about it, bacteria, there's 1,000 pounds of live weight per acre. The Animacetes, 1,000 pounds. We go into the molds, we go into the algae, the protozoas, the nematodes, the insects, the worms. So often we think, okay, our roots are what are giving us our stable carbon or giving us our biomass.
Hmm.
Yes, partly true. And
Absolutely.
through the exudates, we can say, yes, a living root is
Hmm.
building these microbial communities, which is building thousands of pounds
Hmm.
of stable carbon within that soil environment. And it's actually really exciting because that's really what we always talk about. Building soil health comes down to a living root in that soil environment, but an actively functioning living system.
Yeah. Maximizing photosynthesis, which is allowing more carbon to be extracted from the atmosphere, utilized by the plant for growth and reproduction, but also massive quantities are sent down to the soil to generate soil, to make those microbes have the ability to grow their homes. And we're going to hear a little bit from Dr. James White, and a big thank you to Think Regeneration for sharing some of this info with us.
Because what would have happened in the real world is you would have had microbes from internal and you would have had things immediately coming from outside. And in fact, some of the things from outside even have an effect on the germination rates of the seed, improving germination rates. So we know that they're influencing early germination.
And we see that on a very regular basis. We know that these systems are connected. And we know that when we have a lack of something, like we were just talking about, if you have saturated soils, if you're lacking gas exchange, that's going to decrease the capacity of that living system to function. So when we're looking at these biological systems, it starts right from the beginning. It starts right from the seed, or theoretically from before the seed. Whatever we did to the parent plant and the microbiome that parent plant had that are deposited in those seeds make a difference. The problem is it's hard for us to have a lot of control on how our seeds are produced unless we're saving our own. And we do see benefits when we have season after season of saving one seed and replanting, because what you're doing is you're selecting for organisms, you're selecting for health traits that are associated with your specific growing environment. But we can't always do that. So what's an easy way that we can kind of overcome some of these
Well,
basic issues?
and a lot of times, like you say, fallow ground or whatever the scenario might
Yep.
be,
Yep.
let's make sure that biological community is right around that seed as a seed treatment,
Mhm.
as an in-furrow application, as Dr. White's to express those benefits of that point right at germination,
Yeah.
which is critically important. You know, when we start to think about it, most of our seeds these days come with a coating of
Mhm.
it insecticide, a
Yep.
fungicide, whatever, which shuts down some of that natural biology on the outside of that seed. So let's make sure everything that's there so we enhance that germination and that root
It starts
development,
off
that root,
compromised.
yeah, that root morphology. I mean,
Mhm.
we've talked a lot about that when it comes into plant expression.
And we see here, this is New Mexico 2024, and we can see they were utilizing biogenesis, a little bit of humic acid, humic powder. And if you look at this soil in the background, this is dry. This is basically sand. It's less than 1% organic matter. And then we look at this soil that's been generated right around that plant, right around that root. We can see that it has much higher moisture holding capacity. We can start to see some of those microaggregates that are formed that allow for better gas exchange, better moisture holding capacity, better protection of the plant. And that's what we want to see when we're not focusing on those organisms that are allowing that plant to have that diverse species density and the diversity of those species to allow extraction nutrition. When we start to do forced feeding, when we start to think that we can shove a vitamin donut in something and make it healthy and happy, we start to see shifts and changes in those roots. The extreme side of that, if we have a completely sterile seed, it doesn't even form root hairs. That is the extreme end. So we have to have those organisms present for that digestion to start. And I love your example when you were in Europe, and I love how that kind of connects, and we can think about this idea of extraction, but also the surface area that we have associated with that zone around the plant.
Well, yeah, and when I was in Europe, I saw this is the freight map. Basically, if I wanted to go to France, from France to Portugal, I had one way to get there. And that's kind of what we see with our root development in conventional agriculture, which leads to this peach tree of a express nutritional deficiency. We are not mining all of the nutrition out of that soil environment because we have limited access.
Yeah.
We're not seeing the entire country. For an example, when you go to the commuter train system, I can see the entire part of Europe. However I choose to get from France to Portugal, I can do that. I mean, I can see the wines, the breads, the ports all across that country. And I get all.
You got one thing on your mind there, buddy.
Well, it sounds like I'm drinking a little alcohol between that. But no, I mean, really what it comes down to, any food that I want is available to me across that entire country. Which really comes down to when we start to talk about nutrition, water holding capability, soil health, carbon, polysaccharides, glycine, all these things to build soil health. And if you think about it, which is a quicker way to build soil health and diversity in our microbial community? If we go back to the freight train system, where we have one route where we don't even access most of that soil. So we're not building any of these things in all of the other parts of Europe, as if, for an example, on the freight train system, we're seeing that entire country. We're building structure, soil health, and that's really what we see on these routes down here on the far right side. I mean, a community that is putting exudates into that soil environment, feeding diverse microbial communities within that soil environment, which is really what we want to do if we're talking about generating soil health and carbon.
And carbon is what makes up life. All of the organisms that we know of, all life is based on carbon. So carbon is this key link. It's this key building block. I think it's Legos. You can build anything out of it. And that's the importance of what we see associated with it. And microbes recognize that as well as our plant systems. So what we see is our assumption, we look at these roots, we look at our field debris, we look at our active, our passive blankets, and we think about, that's it. That's most of our organic matter. But the reality is that's not the case. Even though living biomass of microbes is small, these organisms grow, live, and die at a rapid pace. This means that microbial inputs to soil organic matter can be much larger than previously thought. We show that microbial necromass, dead, can make up more than half of the soil organic carbon. Hence, we suggest that next generation field management requires promoting microbial biomass formation and necromass preservation to maintain healthy soils, ecosystems, and climate. That is the future. What we are finding from research now is the only way we're going to be building our soils and not reducing our soils, which is what we're seeing right now is massive erosion, massive loss of soil. We look at the dead zones in the ocean. That can be reversed if we start looking at this idea of, okay, let's take that carbon, let's stabilize that carbon and build it into these microbial necromasses, whether we're talking about the extracellular polysaccharides generated by the bacteria that are binding those individual soil particles together. We look at the glomalin that's produced by the mycorrhizal fungi that's taking some of those microaggregates and creating macroaggregates. That's where we're building soil. And we see in our cropland, 51% is associated of our soil organic carbon directly associated with necromass. Our grasslands, 47%. We get into a forest, and we typically think about a very rich, earthy soil of our forest, and we think about all that plant matter, all those leaves, all that material. It's still 35% of that entire environment is necromass. It's those microbial bodies.
So, I mean, when we talk about agricultural farming, most of that is in between the cropland and the grassland, somewhere in that mix of developing what
succession.
we're looking at for creating succession,
Mhm.
either through that building organic matter and stable soil, or working backwards back to rock.
Yep.
Is what it comes down to. And it doesn't matter whether it's sand, rock, soil without structure lacks
It's
water.
dirt. It's dirt.
Yeah, it lacks all the abilities that we need.
So how do we know what is happening in our soils? There are different ways that we can look at our soils and evaluate. We can take a look at our plants. We can visually assess and see what's going on, like we saw that peach tree. But by the time you get to that level, there's already been significant yield loss. There's already been significant damage, and we have a much higher likelihood of insect and disease coming in to take care of those plants, because Mother Nature has started to recognize those as they're not food anymore. They need to be taken out. So how do we assess and how do we evaluate what's going on?
We always, as we've always said, test, don't guess. I mean,
Mhm.
that not only is important when we come to overall plant health, but it's also very important based off of overall soil health, based on everything that we just talked about, because that plant needs to be functioning at a very high level in order to feed those organisms within that soil environment. So we say we need a soil test. We need to know where our foundation is and where we start. But then we have to look at our plant sap analysis or leaf extract analysis. What is available in that soil? What is that plant actually utilizing and getting out of that soil environment? And then a lot of times we talk about our inputs. I use compost here. It's a great example where we're putting out compost because maybe we want organic matter, we want more nitrogen, but we're bringing in tons of sodium, aluminum, chloride, excessive phosphorus, excessive potassium. So it's junk food. It's not what the plant actually needs. And so remember, we talk about with a good, healthy soil, it can supply basically the food, the nutrient, the water, all of these things that the plant needs. But we have to understand how they all function and how they're interconnected. Each one of these, when we look at it based off the test, is interconnected to the other one. And we don't even have a water test up here. We need to add a water test. How we apply our products and where our bicarbonates, where our pHs are within that are critical based off of nutrient availability to that plant and how that nutrient functions within that environment.
Well, I like that idea of looking at compost, and compost can be fantastic. Compost can be a fantastic addition to what you're doing. The other side of that, though, is we have to have an evaluation. We have to know what we're applying, because if we're bringing in things that we don't want, we're all of a sudden forcing the system in a specific direction, and we start to have excesses. And you oftentimes say excesses can be much worse than deficiencies. So when we start force feeding some of these foods, we see a shift in the plant. And like we said, we're going to beat up nitrogen. Nitrogen is basically, it's junk food. It is synthetic food. It is looking at kind of this American diet where we're taking these ultra processed foods and we're shoving them in, and they're full of things that our body really doesn't need. And we have consequences associated with that. And we're seeing the same thing is happening in nature when we start force feeding the plants a material that it doesn't need nearly as much of as we think it does.
Well, and a lot of times I look at when we talk about nitrogen, form of nitrogen is critically important. We see that basically the rhizophagy cycle shuts down with excessive nitrogen in that soil. And, you know, I always say this, so what is excessive nitrogen? A lot of times that has to do with the crop that we're growing, but it also has to do with, if you go back to a soil analysis or what we're feeding, what other things are there in order to convert that nitrate or that amino form of nitrogen. So a lot of times we can have soil deficiencies and we have to be at a low level of nitrogen just for the ability to utilize that nitrogen within the plant and convert that to basically proteins or something that the plant can actually use.
Our goal is balance. And as soon as we force that system out of balance, we're opening it up for attack. We're opening it up for disease. We're opening it up for shifts and changes in the microbial communities. And that's kind of an interesting thing that I want to share. This tidbit from Dr. White about when he's talking about how do these systems function, what happens with nitrogen applications.
Probably a technical question, but in your experiments, are you starting to understand how the plant communicates to these bacteria? You know, you mentioned that they will actually leave with some type of signal to go retrieve a nutrient to bring it back into the plant. Do we understand how that is actually happening?
Yeah, that's not complicated. It's not complicated. It really is quite simple. Plants, at least in terms of nitrogen, if you think about it, when plants lack nitrogen, what they do is they withdraw nitrogen from their exudates. So they don't put out any nitrogen. Instead, they put a bunch of sugar out and organic acids and so forth, but no nitrogen. And what that means is that the bacteria who are able to grow there, replicate and grow there, are the ones that are able to fix nitrogen. And those are the ones they take in. And there's plants are manipulated for other nutrients. There's other, they have other strategies for other nutrients, which is not as easy to understand. And so I'm not going to talk about it, but the nitrogen one is very easy to handle.
And that's where we start talking about this idea of environment dictates expression. If we're creating an environment where that plant has all of the nitrogen, all of the phosphorus, all the potassium that it could possibly ever need, if we're force feeding and shoving these convenience foods down its throat, it doesn't have to work for them. And when it doesn't have to work for them, it's not going to feed the organisms that are required to do it. That makes perfect sense. If a plant needs more phosphorus, it's going to recruit organisms that are better at extracting phosphorus. If the plant needs more nitrogen because the soil environment is not providing it, I love how simply Dr. White puts that. If you don't have nitrogen, the only organisms that are going to be able to grow there are ones that can fix their own nitrogen. So we can start utilizing these systems for our benefit. And this was some weak trials that we did here this last year, where we're looking at trying to isolate and identify different species, different types of plant, different seed types that work better in an environment with less nitrogen and work better with some of these nitrogen fixing organisms.
Yeah, and so like I said, there were 13 different varieties of wheat we looked at, all based off of how this seed was bred. Yep. And how it basically was it going to perform better under a biological or a conventional. And the varieties that did the best obviously were the Hefe, the Trident, and the Shine, which are very popular varieties here in the Pacific Northwest especially. The interesting part about that is we looked at this based off a different nitrogen application. So the conventional program, we had 120 basically all put out early at the time of planting. We had a split application of 120 put out early, and then an additional application top dressed in the spring. And then we had a split application of 80 and 40, which was 120 pounds of nitrogen. In the biological program, we put out 25 at planting and 25 pounds in the springtime in April. The interesting part about this is we noticed that with less than half the nitrogen of the conventional programs, we came out with very similar yields. And that's what we were talking about, is how far, what we were trying to identify here is how far can we back off that nitrogen and maintain yield. And then looking at ROI based off of cost and everything else that was involved in doing this. And the ROIs on all of these were very similar. There was one exception of the ROI, basically, when we looked at the 138 yield based off the 131 yield with the nitrogen fixing bacteria. But then we have to look at not only yield, but we have to look at quality. And how does that compare into basically what do I get paid based off of quality? When we start to talk about protein levels, either high protein or low protein, all of those test weight. But it was really interesting. But, with Dr. White talking about if we are using excessive nitrogen and we shut down that rhizophagy cycle, not only are we not getting the nitrogen, but there's other things we're not getting in there, Steve.
Yeah, and I laugh a little bit because I think back in the 70s and 80s, what was Bruce breeding varieties of wheat for? Limited nitrogen and still maintaining protein, because his focus was how do we feed the plant? We feed the plant with these bacteria. And like you're just saying, it's not just nitrogen. So when we over apply nitrogen, the plant no longer needs to grow those organisms. And the downside of that is we're seeing more than just a nitrogen loss. When we look at a bacteria, the basic profile is a 10-2-2 plus everything else, because all of those organisms, they need silica, they need zinc, they need copper, they need manganese, they need all of those for their own enzymes and for their own life processes. So when a plant is taking these organisms in through the rhizophagy cycle, which is basically it spits out exudates, feeding the microbes. Microbes reproduce, they extract nutrients from the soil, they move into the plant bombards them with reactive oxygen species, superoxide in this case, which breaks down their cell wall. They get swollen, they swell up, they get leaky, some of them die. Oh well, plant eats it entirely, but it also has the capacity to select for those organisms, like I mentioned, like phosphorus. It can detect which organisms has a higher capacity for extracting those nutrients. It clones those organisms and then it spits them right back out. So by utilizing the rhizophagy cycle, we're allowing the plant to feed off of all of that, rather than if we go out and we apply N, P, and K. You know what the plant has access to? N, P, and K.
But if we think about this, Dr. White talks about root morphology and those fine root hairs
Yes.
being created to spit those microbial communities back out into that soil environment.
Mhm.
And if we think back on the commuter terrain in Europe, that's what we want. We want more fine root hairs,
Yes.
more of these exudates. When we start to talk about building stable organic matter, polysaccharide, all those things we talked about building soil health is critical to this process. And when we use excessive nitrogen, not only do we shut down all these other nutrition that we're getting, we shut down the root development within that soil environment.
And so many other things. When we're limiting the organisms that are associated with that root, we start looking at the functions that plant growth promoting rhizobacteria have. We see them producing phytohormones, solubilizing phosphorus, induce systemic resistance. They're producing ACC deaminase. So if we have a drought stress, if we have heat stress, if we have heat shock, so these organisms are protecting the plant from both living biotic stresses and abiotic stresses like heat, like cold, like frost. But also we see a better extraction of nutrition. We see better breakdown of soil, better soil synthesis, like we're talking about with that necromass. There's so many things that we just keep stacking on top when we allow the system to function. Utilizing some of this data that we were looking at in the wheat trial, because we were looking at this from what else do we see associated with benefits from the biology? And we start to see other shifts and changes. One thing that we saw from, this is research or documentation data from biome makers, was a resilience index. Resilience is basically adaptability. So if I have a football team and I take away their quarterback, they're pretty well done. They don't have necessarily a backup, so they don't have a high level of resilience. So if we go into a soil environment where all of a sudden those plants that we're expecting to have all that nitrogen fed to them, they don't have those organisms present that can do that job, they collapse. We no longer have a functional system. When we add these organisms, we saw a 46% increase in resilience associated with the genetic profile of the organisms present in that soil. So we have an environment that, okay, it gets a little hotter, it gets a little drier. It doesn't matter. The plant has a better ability to adapt to that environment. Looking at the nutrition, 29% increase in potassium availability. We saw a 21% increase in magnesium, a 16% in calcium, and a 15% in phosphorus. So we see all of these things like we were talking about. It's 10-2 plus all the traces. It's not just the one nutrient. It's all of these things. And this is looking at the genetic profile of the soil, of the organisms present in the soil. But we also need to go above and see what's actually making it. So we see a better ability of that soil to process the nutrition and feed that plant through rhizophagy. But that's not the whole story. We need to look at the rest of the plant as well.
Yeah, so now we look at basically from apical a plant sap analysis. So we look through there is what was the plant actually uptaking based off of the nutrition, based with a correlation based off what biomakers saw. Is the plant, even though that's happening in that soil, is the plant actually getting it? And when we looked across there, yeah, they had the plant actually had a higher total nitrogen than the applied with 120 or 150
It's
pounds of
just
nitrogen.
it's fantastic to think about a system where we're applying less than half, and yet because the way we're feeding it is the way the plant wants, it is encouraging its uptake. It's just fantastic.
And we saw higher potassium, higher magnesium, higher calcium, higher manganese, cobalt, phosphorus, silica, iodine, boron, molybdenum, and selenium. Across the board, higher in the plant by not only levels, but also percentages, which is a lot of times what we look at.
Yeah.
And the thing I want to mention here that we haven't even talked about, this was as we talk about, a liquid inoculum that was applied in furrow at the time of planting. One application with that seed at germination, and how these microbial communities function and grew like we were talking. A dry seed inoculum, they're creating an environment. The plant is basically relying on them to make nutrition available, and that biology is relying on that plant to feed them so they can reproduce and make the things available to the plant. But it doesn't only happen in the soil. That's the cool part about it.
No, we talked a lot about this idea of the rhizosphere, this zone around the root, but we can also take a look at what's going on above the plant, because believe it or not, the roots are somehow magically connected to the leaves. Did you know that?
I thought there was a disconnect where the soil separated them.
Until I saw this picture, So, joking aside, when we start looking at what's going on in the phylosphere, we see very similar situations going on. Phylosphere colonizing microbes play critical roles in multiple functions, including plant productivity and fitness, affecting leaf function, longevity, seed mass, apical growth, flowering, fruit development. And also key roles in removing contaminants. So basically all these things that we want the soil and the plant to be doing, we can also see associated when we do an inoculation of the leaf. We see better release of nutrients. We see better communication with the plant. We can see direct uptake of phosphorus, potassium, iron, zinc, sulfur, and copper. We also see that that environment is going to be shifted and changed. Just like we talked about the microbes creating a zone around the root, that roster root that is protected, they're creating a home and a house. And we saw that the picture from New Mexico where the soil was less than 1% organic matter and the soil was very dry versus around the seed or around that root, which was a much different environment. We see the microbes doing the same thing. They're producing extracellular polysaccharides to create an environment on that leaf that is allowing them to grow longer. As they grow longer, they're able to extract more nutrition, they're able to feed the plant, they're able to communicate with the plant, and they're able to do all of this because it's a very unique environment on the leaf surface. When we look at it and we think about it, yeah, some of them have a bit of fuzz, and that's about all we can see. But the reality is it's mountainous when you get down to the bacterial scale. So they're creating all of these little micro environments on and around the leaf that allow them to work with the plant, to protect the plant.
Well, and that biology is creating an environment in order to support itself.
Yeah.
That actually benefits the plant. It works as almost a biosurfactant.
Yep. Oh, it
where
does.
It creates a film on
Mhm.
that plant, which creates moisture, which creates nutrition.
Mhm.
We understand with foliar nutrition, we need to keep it wet in order for that plant to be utilized by that plant. All of these things are naturally happening with that microbial community on that leaf structure, which, I mean, we even go back to the last slide and we talk about the idea of, I call it priming the pump.
Mhm.
Foliar nutrition on that leaf structure sends exudates into that soil environment, which gets that microbial community within the soil to make more of that nutrition available that we might have just put on as a foliar based off a deficiency from a plant sap, which is now creating more of that nutrition based on microbial communication.
Mhm.
I mean, it's pretty cool how all this works.
I love this idea of inoculating a leaf, and we're purposefully allowing the plant to recruit specific organisms for its benefit. We see some of these organisms associated with direct protection. So up on the leaf, occasionally there are some pathogens that might attack a leaf. I think we've seen that a time or two in agriculture.
Yeah, and
But-
whether we talk potatoes, whether we talk wheat, whether we talk corn, it's out there. Mother Nature is not nice sometimes.
No, fungal pathogens are everywhere because they're doing a job, just like we talked about weeds or plants growing where we don't want them. They're just doing a job. These are saprophytic organisms that are designed to break down dead and dying tissue. If they recognize your plant that you think is alive and healthy and happy as dead and or dying, they're going to start breaking it down. So the beauty here is the microbes in the soil and the leaf are helping that plant extract nutrients, protect itself, grow, but we're also shutting down the capacity of those pathogens to talk to each other. This idea of quorum sensing. Every microbe is constantly ejecting little molecules, homoserine lactones that are just talking to each other, letting each other know, hey, here I am, there's this many of us, and the fungal pathogens and other disease organisms are doing the same. Microbes that are beneficial to the plant don't want that plant to be harmed. So they're going to purposefully consume those communication molecules. They're going to directly impact the ability of those fungal pathogens to talk to each other and to affect the plant. And I've got one more little clip here that is kind of fun from Dr. White that kind of talks about that.
Anyways, that results in hardiness, increased hardiness and stress tolerance in plants. And that means they're climate change, it means salt, it means heat, it means diseases. The plant gets tougher and hardier. Also soil pathogens, the microbes that are involved in rhizophagy cycle come out of the plants, and some of them will colonize pathogens, fungi in the soil that cause disease, things like damping off pathogens, alternaria, for example, and fusarium get colonized, and the bacteria will go all over the surface, but also go inside the hyphae, and that changes the behavior of the fungus. Fungi tend to be not aggressive, not pathogenic when they have bacteria.
So I love this idea as we're doing all of this. So we're reducing our fertilizer input, which is saving us money. We're then allowing that plant, from what we've seen in the research, to feed itself better. As we increase those bacterial and beneficial organism communities, they're also colonizing the fungal pathogens and basically shutting them down. They no longer have the capacity. So we're never going to get rid of them. They're always going to be there. Life is everywhere. Like we just were talking about in the beginning, we see microbes on the leaf, on the seed, in the fruit, all over the place. They're always going to be there. One of the capacity of those microbes is protection. And I like this little picture. Microbes, they're just wearing a whole bunch of different hats. So we look at this EPA biopesticide list, and we see a wide diversity of organisms that are associated with protecting the plant. We can see direct impact associated with fusarium, Azospirillum braziliensis, Bacillus subtilis, Pseudomonas fluorescens, Trichoderma harzianum. We can see Phytophthora, where we have a group of organisms that are protecting the plant. Powdery mildew, Rhizoctonia, Verticillium, basically all of these organisms that are trying to harm the plant, the beneficial microbes want to protect the plant. And I love this little video looking at what's going on the surface of a leaf. So this is Pseudomonas syringii. It's a motile organism. It's moving around, and that's inside the stomata. That plant is now, there goes another one. That plant is now infected with Pseudomonas syringii. However, when the researchers treated the plant with a probiotic, in this case Bacillus pumilus, it allows that plant to sense those Pseudomonas syringii and shut their stomata, no longer allowing those syringii into the plant so they can no longer infect and they can no longer damage that plant. There is so much communication going on. There's so much interaction going on. And we can see that with more than just the fungicidal organisms. There's bactericidal, like we just saw with the Pseudomonas syringii, nematicidal, insecticidal, antiviral. Basically, any way that the plant is going to be attacked and decrease its ability to photosynthesize and produce carbon that feed those microbes are going to protect the plant from.
Well, and it's, we give the example, I love this, it's the bat signal.
Yeah.
The plant sends out the bat signal, and basically the microbes are Batman and Robin. They're going to be the pal, the cabal, they're going to basically help suppress or basically quiet that disease pathogen, that organism, because that's what it's designed to do. If the plant can function as designed. Bruce used to say, pest and disease do not arise because of a deficiency of pesticides in our environment. They're simply a symptom of poor nutrition.
Yep.
I mean, soil and soil nutrition, plant nutrition.
Yep.
That's really what it comes down to. And if we don't have these organisms and we don't have a balanced, healthy plant, we're not making that nutrient available to the plant. We're not utilizing our phyllosphere or our foliar applications
Mhm.
based off of information testing and don't guess. We get to head down what I call the aisle of death. We get to go down the aisle of death, and then we have to spray these. And the most amazing part about this is we don't really talk about that. There is a ton of research, and oh yes, it's mentioned here, there are side effects when utilizing these, not only to the biological world on that plant
Mhm.
or soil health, but by that microbial world, but also how that plant reacts.
Yeah.
Once that plant is sprayed with a fungicide or an insecticide, or even we'll talk GMO crops where they're sprayed over the top with a Roundup, glyphosate, Roundup Ready crop. They go through this process where they now basically get sick.
Yeah, they're
They've got a
stressed.
light dose, they're stressed,
Mhm.
and now they have to do something to heal themselves, which takes energy, which reduces overall plant health, basically in order to combat something that we're doing as a beneficial. So really, it comes down to, I mean, it's you are what you eat,
Yeah.
as we talk about.
And Charles Mercier, 1919, way back then, the work is with living things, and living things cannot be managed by coercion. They must be understood. And that's where this idea that Bruce talked about and we talk about regularly, everything is interconnected. So if we're going to allow our systems to function at their maximum capacity, we have to utilize natural systems.
Yeah, we have to utilize microbes. We have to look at the soil. We have to look at the plant, the animal, the human component. All of these are interconnected, and how do we manage those to basically the optimum level based off the crop that we're trying to grow? And with
Awesome.
that, I'd like to thank everybody.
Thank you.